genetic mutations. These are going to be permanent changes to DNA. So heritability, the mutations are going to be happening to a cell's DNA. If that is a somatic cell, then again, this is sort of a body cell, so normally a cell that is already part of a body, and it is going to be maybe producing new cells, copies of its own, but it is not the cells that create an entire organism. So those are somatic cell mutations, and this can happen through mitosis. Germ cell mutations, though, this is going to be affecting meiosis. So this would be the development of the germ cells, the gametes, so eggs or sperm. If they have a mutation in them, then any individual that arises from those, every single cell in their body has been derived from those germ cells, and therefore every single cell in their body likely could have that mutation. So the different types of mutations, we'll go through them. These ones here are point mutations, so they are changing a single base pair, so one of the letters in the DNA code. It could be a substitution mutation. The names kind of indicate what's happening with it. Substitution mutations where one nucleotide switches for another, and as we know from our codon table, depending on what nucleotides end up in the mRNA from the DNA, you're going to get particular amino acids. Sometimes if you switch those, you get a different amino acid. Now remember the third letter of the codon is like the wobble one, where several, like say leucine here, CU anything will give you leucine. So if you change the last letter, it's not such a big deal, but if it happens to be the first letter that changes it, then you would get something very, very different. So CUU would give you leucine, but let's say you had let's go with UUU, you'd get phenylalanine, I think that is. It would be a different amino acid, and therefore the protein that is made with that different amino acid may function differently. They could be silent, and this is again those wobble effect ones. If it's affecting the third letter, it's going to be a silent mutation. And again, example here with glycine, if it's the third letter that's changed from a C to a U, yes, that is a mutation, but it is silent. It does not have an effect. It does not change the amino acid, and therefore there'd be no change in the protein itself. If it is a missense mutation, it's going to make an altered protein. And so, for example, if you switch from GAA to GUA, you're changing the codon to the point where you get, instead of glutamic acid, you get valine, and so you actually do change the protein itself. You get an altered protein. And then a nonsense one would be where you don't get a protein. So missense, you still get a protein, it's just different. But a nonsense mutation, you actually get something like a stop codon showing up right in the middle of the protein, and therefore the protein itself is going to not get finished being made, so you don't even really have, it's not like it's a slightly different protein, it is not even a protein. Missense mutations, for an example with this, it would be classic example would be sickle cell anemia, and again it is just a single letter that is mutated. And if you look at the code, this one particular codon that makes this protein here for hemoglobin, a big protein, lots of amino acids in it, one single codon is changed by one single letter, so a point mutation, and this is a missense one because it makes it puts down valine instead of glutamate, and the hemoglobin itself is going to have a function. It's still a hemoglobin protein, but it doesn't work in the same way, and then this is what leads to sickle cell anemia. Frameshift mutations is where a single base pair is added or deleted. And this is going to cause, again, because you're doing if it's a single addition, you're going to cause an entire change to the reading frame, and therefore the protein is completely unlike the original protein. So we would say this is a nonsense mutation. You do not get anything close to the original protein. So an insertion would be adding one. And so again, if we added a C right here, you'll see that what was GGA is now CGG, and so you end up with a completely different amino acid there, but also you're never going to get the GGA again because the reading frame is off. It has changed because of this insertion of this one nucleotide, and therefore everything downstream from there is going to be making different amino acids, most likely. Deletion would cause the same thing, and so if you get rid of this G here, then everything shifts to the left by 1, and therefore everything downstream of that mutation that is being read will give you different amino acids, and therefore you get a completely different protein, and wouldn't really be a protein because it wouldn't really do anything. So two opposite frame mutations can actually cancel each other out. If you had a deletion and insertion, even if they're not in the exact same spot, they'd probably only result in a small change. Even if you get a couple of different amino acids, it normally may not have a drastic effect. But we do see sometimes it does with sickle cell anemia. But at least you'd have most of the amino acids in the right spot, and so you may still have a somewhat functional protein if you have two opposites. Or if you had 3 insertions, that would do it too, or 3 deletions, that would do it too. Chromosomal mutations, so these are much larger scale, not point mutations, and so there are several things that can happen with chromosomes. They're being moved around as the cell is going through mitosis and meiosis, and so if this happens not in the normal way, we get what's called a non-disjunction. So there's an uneven splitting of chromosomes during meiosis. And so here we have metaphase, they meet up and they're supposed to split evenly to either side of the cells. But in this case here, we've got the entire chromosome going to this one side, and so this cell here is going to be missing a chromosome, and this cell here, when they're derived here, again, they do one more split before they're done, we're going to have extra chromosomes. And so this is a non-disjunction event. Translocation is where you actually have movement of DNA sections between different chromosomes. And so here we have the original chromosomes, and sometimes an entire chunk of the chromosome so this bottom chunk and this bottom chunk can switch places. So we can have translocation happening within those chromosomes. Inversion is kind of like translocation, but instead of moving from one chromosome to the other, the piece itself actually is cut and then sort of moved around within the same chromosome and actually gets split around into a different orientation. And you don't have a single change to a particular protein, because remember, within these chromosomes, the very small section of them will be encoding the genes, and so these may not affect how the genes and therefore the proteins are actually read, but it does change the location of them within the actual chromosomes. And again, if you think of translocation, this is one of the things that happens regularly during meiosis. Transposons are genes that, so the segments of the DNA that actually change position in the chromosome. So instead of an entire section, like with inversion or translocation, it could just be a particular gene or set of genes or small location of DNA that is actually moving its spot within the chromosome. And again, as long as they still function, it's not going to cause any deleterious effects because of it, most likely. And so again, there's a last one here, fusion of chromosomes, and we've seen this. If you look at the karyotype of a chimpanzee and the karyotype of a human, there's some similarities there, and one of them looks as if there has been a fusion of chromosomes. We actually have one set fewer than chimpanzees do, but it looks like one of ours is actually made up of 2 of them that have been fused together. So here's some analogies to go over those types of mutations. So substitution would be like if you had this sentence here, Sam now arriving from Guyana, and you change the W to a T, you can see how you can very quickly change the meaning of the sentence itself. So that is like a substitution mutation, one letter changing for another. A deletion would be, again, could cause an effect depending on where the deletion is taking place. If it's in a gene, it's going to have a big effect. It changes the reading frame. Insertion, same thing. If you change the reading frame, you can change the meaning of it. Translocation here, where you have the whole section moving from one place to another. Or an inversion, where you have a particular portion of the chromosome flipping around and reinserting itself, and again, may not have a particularly deleterious effect if those sections within it remain intact. So the causes of these mutations can be spontaneous. Again, things can happen as the DNA is copying itself and it may make an error with it. A lot of times it would be corrected, but occasionally it is not. One of those errors could be that a nucleotide could be added without a nitrogenous base to it. And what will happen is when it's trying to when the DNA is trying to be copied, so we have this is the original strand here, and if while it was being copied, you ended up with this side here not getting its nitrogenous base, then the next time it gets copied, what our DNA polymerase is going to do is it's going to say, okay, I don't know what was supposed to be here to match to. If it was a G, they should have put a C there. But if there's no nitrogenous base there, what it will do is it'll say, okay, well, let's just go with A. It's like when you're guessing on a multiple choice test. I don't know what the answer is. I'll just go with A, which is not a bad choice, but again, it could be wrong. And so what will happen is then in the next round of copying, when this strand gets copied from the one where the A was just randomly picked, you will end up with a thymine going there. And so what we have happen, and again, it could be correct a quarter of the time, just going with A. If you're telling you 4 choices, you're going to be right a quarter of the time. But what happens is if you always are going with A, as DNA polymerase is, you're going to end up with a extra insertion and an increase in the amount of pyrimidines. You're losing these purines because the default, the guess that DNA polymerase is making is to go with A. So that is a spontaneous mutation and how DNA polymerase deals with it. This causes the loss of the purines. Induced mutations can be caused by external mutagens. So again, substances and energy. So let's first look at physical mutations. And so these are like X-rays, the gamma rays, all the superhero backstories. They can change the DNA, and they do this by essentially affecting how it is bonding. And so if we look at these thymine dimers, they're called. And so again, these nitrogenous bases are supposed to be hanging out next to each other. We have one T beside another T, and they're not supposed to be bonded to each other. They're supposed to be hydrogen bonded to their complementary strand. They're supposed to be covalently bonded to the sugar phosphate backbone. But UV radiation can actually cause them to form these bonds between them, and this is going to warp the DNA and change how it is going to be able to be read. And so what happens is you get these T's that are bonded to each other, and you end up with this changed DNA. It's not going to be able to be copied or read the same way. Chemical mutagens, as we have seen lots of examples of them, so it sounds like smoke and diesel fumes and all that, they can cause essentially act like the DNA bases themselves, but they don't pair properly. So where these nitrogenous bases are supposed to be going, you have something else there instead, and then this is going to lead to your cells trying to have to deal with reading something that's not actually correct. It's not actually made of the proper bases, and so this is going to change how the DNA is being read, which is going to be a mutation. They can also bond to the bases themselves, so not just replace them, but actually sort of mess with how they are interacting, so they can bond to them, and that way is going to change the shape of the DNA, again affecting how it's going to be copied and read. They can cause these frameshift mutations, so actually getting in there like a whole nitrogenous, or like a whole nucleotide, and causing a frameshift mutation. Things like benzene can do this, and we refer to them as carcinogenic because they may lead to a cell having uncontrolled growth. That's what cancer is. Viruses also are able to cause these mutations to happen. So they, again, work by trying to get our cells to make things for them by inserting their, not just DNA, but RNA as well, into their host cell. And this can interrupt the cell's genes. And so herpes virus and HPV, human papillomavirus, can cause this to happen, and they actually lead to the development of cancer. Repair mechanisms for this, we have luckily a number of them because our DNA is constantly under attack. And so there is direct mutation where you have DNA polymerase, which is able to sort of find these errors or mismatches within the DNA and replace them. Excision, the mutations are removed after the fact. And so we have the enzyme going in there and cutting them out, and then them being replaced by DNA polymerase. And a recombination where you have, again, excision would be for one single strand. We still have the other one that you could use to be read to find out what the proper message is. So if you cut out a T, then you know, okay, looking over at the A, I know a T has to go back in there. So that can happen with the excision. With recombination, you have both strands being corrected after the fact. And obviously, if you are cutting out and replacing both strands, you don't have a template to go off of, except all of our chromosomes comes in pairs, except for Y. And so those homologous chromosomes can actually be used to repair that double-stranded portion. If the damage is too severe to repair, again, we've got trillions of cells, so the right thing to do for the cell, if it is beyond repair, might be to prevent itself from growing uncontrollably. And so our cells themselves can, and quite often do, actually break down themselves. They destroy themselves in order to, if they are unfunctional or not able to be repaired, then probably letting the other cells take over for them and them themselves no longer existing may be the way to go.